Excuse me. Okay. So I've shared my slides. Can one of you unmute and let me know if my presentation is visible and if my voice is audible? Okay. Okay. Thank you. So good evening. Today is the 10th live session. So it was basically the replacement live session from last week. And as we know that in these live sessions, we basically talk about some questions, some old assignment questions from the school genetic engineering theory and application from by the course instructor Professor Vishal Trivedi from IIT Guwahati. And in this live session, we basically talk about some sample questions and we try to delve into the details of those questions so that it's easier. Try to learn the concepts behind those questions so we can answer not only these questions but similar type of other questions as well. Okay. So having said that, we'll start today's session with the first question for today, right? So the first question for today is cell division occurs in which phase of the cell cycle? G1, S, G2 or M? I'll wait for a few minutes. Can anyone try?
M. Option D.
Okay. Yeah. So yes, you're right. The cell division occurs in M phase, a mitotic phase or whatever. But the other phases are also equally important, if not more. And we'll discuss about all these cell division phases in this question. So, what is the key concept? What problems does growth cause for cells? So, why do cells divide rather than continue to grow indefinitely? So, have you ever thought of this? Any of you? Why do cells divide rather than continue to grow indefinitely? They can get bigger in size and bigger, but why do they choose to divide? Anybody? You can think about it. Okay, so larger the cell. Yes, yes.
Ma'am, actually it is a matter of surface area
Yeah,
ratio.
yeah, right.
That
So
ratio is to be maintained. If it is
correct.
bigger, cannot
Correct.
accommodate.
Correct. So the larger the cell is, more the demands are placed on the cell's DNA as well. Okay. It's more difficult to move enough nutrients and waste across the cell membrane. Okay. The surface area is to volume ratio is what exactly you were saying. So even that ratio needs to be maintained. When your cell gets larger, your surface area will increase definitely, but your volume will also increase. So with such a big volume bundle, it's that surface area increase ratio might not be even, right? Like the increase might not be even enough. Surface area increase might not be even enough to support that huge volume. Then DNA overload, DNA controls cell function and it's found in the nucleus of eukaryotes. So the DNA meets the needs of the cell when the cell is small. However, as the cell increases in size, the DNA cannot meet its needs anymore. Okay? Anymore. So there's an information crisis, right? Because DNA stores information in the form of gene. That gene ultimately gives rise to codons and then protein and amino acids and proteins. So basically DNA is the information hub of the cell. So the DNA can meet this needs of the cell when the cell is small. But if the cell increases in size after a particular limit, the cell DNA cannot meet its needs anymore. That's why there is an information crisis. Then food, oxygen and water, they enter the cell through its cell membrane. Waste products leave the same way. So rate of this exchange depends on the surface area to volume ratio. Exactly what you were saying and what I explained as well. So this rate exchange depends on the surface area to volume ratio. So to obtain the ratio of surface area to volume, divide the surface area by the volume obviously. So you can see the cell sizes, surface area, volume and ratio of surface area to volume. For surface area 6 cm cc, volume 1 cc, your surface area to volume ratio will be 6:1. For 2 centimeter height, width and breadth, height, width and length cell sizes, it will be 24 cc for surface area, 8 cc for volume and 3:1 ratio of surface area to volume. And for even bigger cells, if you see 3 centimeter height, width and length, 54 cc will be your surface area, 27 will be a volume, and your ratio even goes down to is to 1. Right? So you see the volume is increasing so much, your surface areas will not be capable of handling so much, handling the needs of such a huge volume. So exactly what you were saying, the ratio needs to be maintained. Right? So the notice that the volume increases much more rapidly than the surface area. So this causes the surface area to volume ratio to decrease, which is a serious problem for the cell. So as cells get too large, it makes it more difficult to get sufficient amounts of oxygen and nutrients in and waste products out. Correct? But how does an organism get bigger if the cells that it is made of it do not get larger? It grows more cells, right? So how does it get bigger? How does one organism start as a small uh, thing and then get bigger? Because it grows more cells. So what is the division of the cell? Before the cell gets too large, it divides into two daughter cells, and this process is called cell division. So cell division solves the problem of cells getting too large by increasing size while reducing volume, because more small cells instead of one large cell is still less volume and increasing surface area, right? So before cell division, the cell replicates or copies its entire DNA, and this solves the problem of information storage, because each daughter cell gets a complete set of genetic information, and each cell has an increase in the ratio of surface area to volume that allows for more efficient exchange of materials with the environment. Okay, so all cell division must involve the replication of DNA before cell division, so that the genetic information can be transferred to the daughter cells. Now in prokaryotes, the rest of cell division is simply to divide the contents, and in eukaryotes the division is more complex. It occurs in two stages: mitosis, division of the nucleus, and cytokinesis, division of the cytoplasm. So actually it should be karyokinesis, this should be karyokinesis. Karyokinesis means division of the nucleus, cytokinesis means division of the cytoplasm, and the entire process can be called mitosis. So unicellular organisms use mitosis and karyokinesis and cytokinesis to reproduce, right? And this is a type of asexual reproduction, producing two identical daughter cells from one parent cell. Right? Now multicellular organisms use mitosis and cytokinesis to add new cells to the organism for growth and development. Now what are chromosomes? You know, chromosomes are made up of DNA and proteins, basically basic proteins, histones, and they carry the genetic information in the eukaryotic cells. Now each species of organism has a specific number of chromosomes. Like Drosophila melanogaster has 8 chromosomes, humans have 46 chromosomes, and chromosomes are not visible except during cell division, known as chromatin, when they are not dividing. So replication or copying of the DNA occurred before cell division. Therefore, every chromosome is actually two identical sister chromatids. Now, each pair of chromatids is connected to each other at an area called the centromere, and that is usually located near the center of the chromosome. Now, cell cycle, if we talk about cell cycle, it's basically the series of events the cell goes through as they get separated, they grow and divide, get separated by periods of in-between time called interphase. Now, during the cell cycle, the cell grows, prepares for division and divides to form two daughter cells, which each of which then begins the cycle again. And 4 phases of the cell cycle are there. Now G1 phase, basically what happens in G1 phase, cell growth, increase in size and synthesis of new proteins and organelles. S phase, chromosome replication, synthesis. G2 phase is shortest of the phases, organelles and molecules required for cell division are produced. M phase is mitosis, basically karyokinesis and cytokinesis, totally called mitosis or M phase. The G1, S and G2 are all taking place during the interphase, the phase between the divisions. So this is how the entire thing is. G1 phase is mostly for cell growth. S phase is for DNA replication. So you see why I said these phases are even like mitosis is important, but these phases are also important, even more so than mitosis, because the main thing of DNA replication happens in S phase. G2 is basically the preparatory phase before mitosis, and then we have the M phase, which has prophase, metaphase, anaphase, telophase, and cytokinesis, right? So the mitotic phase can be subdivided into 4 phases: prophase, metaphase, anaphase, and telophase. And mitosis is strictly nuclear division, and it is followed by cytoplasmic division or cytokinesis to complete cell division. and it results in two daughter cells which are identical to each other and is used for growth and asexual reproduction. So the growth and synthesis phases are collectively called interphase, and the only source of genetic variation in the cells is via mutations, right? So what is the interphase stage between division? This is when the cell is not dividing but is carrying out its normal cellular functions. So the chromatin is not visible, DNA, histones and centrioles all replicated. Replication of cell organelles like mitochondria occurs in cytoplasm, and G1, S and G2 phases are occurring. Then chromosomes condense. What happens in the prophase? That is the beginning of the mitosis or the stage, kardiokinesis. Chromosomes condense and become visible. Now, due to DNA replication during interphase, each chromosome consists of two identical sister chromatids connected at the centromere, and the centrioles move to opposite poles of the cell. Nucleolus disappears, spindle fibers begin to form and attach to the chromosomes near the centromeres, and the phase ends with the breakdown of the nuclear membrane. Then you have the metaphase. Spindle fibers connect centrioles to chromosomes, and chromosomes align along equator of the cell. Then we have the anaphase. Basically, centromere split allows sister chromatids to separate. Chromatids move towards opposite poles, centromeres first, creating a V shape. Then in telophase, the spindle fibers disperse. Nuclear membranes form around each set of chromatids. Nuclei reform. End of nuclear division. Okay. So this is the entire schematic of cell division. Centrioles are here, chromatin is here. If you see, this is the nuclear envelope. Then centrioles and centromere have divided and are moving apart. The chromosomes you can see. Centrioles are at the opposite ends. The chromosomes have lined up. This is along the equatorial or the metaphasic plane. This is called the metaphase. Then centromere breaks and the basically the chromatids, sister chromatids move to the opposite ends pulled by the spindle fibers. This is called the anaphase, and then the nuclear envelope reforms and everything goes back to the original way, telophase. Now in animal cells, a cleavage furrow, what happens, this is the kardiokinesis, that is the nucleus has been separated but the cells are not separated yet. So this is followed by cytokinesis, which is cell division, the cytoplasm division. So in animal cells, a cleavage furrow, if you see over here, forms which splits the cell into two. Whereas in plant cells, the vesicles move to the equator, line up and fuse to form two membranes called the cell plate. A new cell wall is laid down between the membranes which fuses with the existing cell wall. Okay, so cytokinesis is the division of cytoplasm, end of the cell cycle, production of two identical daughter cells, different in plants and animal cells. Okay, you see over here. Now, how is cell cycle regulated? How are cancer cells different from other cells? So, multicellular organisms control cell growth and division very carefully with this way to increase the number of cells and size of organism. So, this provides a replacement of cells that wear out or are broken down. So, cells in a petri dish will continue to divide until they come into contact with other cells. When the cells stop dividing, cells are removed from the center of the dish. Then the cells bordering the open space will begin dividing until they have filled the empty space, and the controls for cell growth and division can be turned on and off, and we can see the same thing happening in our own bodies. Okay, so what happens exactly when you cut your finger or break your bone? So the cells bordering the injury will begin dividing to fill in the gap in the tissues that have been torn or broken, and this is the process known as healing. So scientists wondered what's controlled cell division. So there were scientists Tim Hunt and Mark Krishner discovered that cells in mitosis contained a protein that when injected into a cell would cause the formation of spindle fibers. So these protein groups were known as cyclins. It regulates the cell cycle and they rise and fall in time with the cell cycle. So cyclins regulate the timing of the cell cycle in eukaryotic cells, and there are two main groups of protein regulators. The internal regulators respond to events inside the cell. That is, they make sure cell does not enter mitosis until chromosomes have all replicated. Then the external regulators that respond to events outside the cell, like embryonic growth and healing. In the case of cancer, which is uncontrolled cell growth is so controlled because when it is not controlled, things go very, very wrong, right? Cancer is a disorder in which some of the body's own cells lose the ability to control growth. So they do not respond to the signals that regulate the growth of most cells, and when cells divide uncontrollably, they form masses of cells called tumors that can damage the surrounding tissues. And these cancer cells can break off and spread throughout the body, disrupting normal activities and causing serious medical problems or even death. So as we come to it, the cell cycle multiplies cells. Basically, it is an ordered sequence of events that extends from the time a cell is first formed from a dividing parent cell until its own division, right? and it's a continuum of dynamic changes. During cytokinesis, the cytoplasm is divided into separate cells, and the process of cytokinesis differs in animal and plant cells. So in animal cells, an equator ring of actin filaments constrict, plasma membrane starts to sink in, forming cleavage furrow. It's called the contractile ring mechanism. In plant cells, vesicles cluster and fuse, two new membranes lengthen, and cellulose is deposited between them. Cell walls begin to form, called cell plate formation. Okay, this is how it happens. Now, anchorage cell density and chemical growth factors affect cell division. Cells within an organism's body divide and develop at different rates. So, cell division is influenced externally by the presence of essential nutrients. Growth factors, proteins that stimulate division. There are over 50 different growth factors which work for one or more cell type. Then density dependent inhibition, in which crowded cells stop dividing. Anchorage dependence, the need for cells to be in contact with the solid surface to divide. Now, the cell cycle control system is a cycling set of molecules in the cell that triggers and coordinates key events in the cell cycle. To checkpoint the cell cycle can stop an event or signal an event to proceed. Now, there are three major checkpoints in the cell cycle. Three major events in the cell cycle. The G1, commitment to divide, growth factors present, size of cell is okay. Check for proper DNA replication, G2, and all chromosomes attached to spindle fibers in the M phase. Right? Cyclins are proteins produced by the cell during cell division. Cyclin dependent kinases or CDKs. Cyclin is required to activate these enzymes, activate cell proteins by phosphorylating them. Proteins needed for S phase, needed to go through G1 checkpoint. Then we have the MPF, maturation promoting factor or mitosis promoting factor, with cyclin CDK complex, phosphorylating proteins needed for mitosis needed to go through the G2 checkpoint. Now, rate of cell division, it differs from one cell type to another. So, red bone marrow cells divide every 12 hours to replace RBCs that wear out. Cells at tip of root divide about every 19 hours. And neurons and nerve cells normally never divide again once brain is fully formed in utero. So, if the control of division is lost, it gives rise to cancer. So, cancer is different depending on the tissue affected. Common theme is lack of control over cell division, abnormal uncontrolled cell division, mutations in genes including p53 that target and control abnormal cells. Abnormal cells impede functioning of normal cells, right? So what is the p53 gene? It's a tumor suppressor gene. It has a key role in G1 checkpoint. p53 protein monitors DNA. It's found absent or damaged in most cancer cells, right? So if you see with p53, cell will repair or die. Like mutation will repair or die. Without p53, cell may just go mutation crazy and start dividing, right? Dividing without any control. Okay, so tumor suppressor genes, basically the name is self-explanatory. It prevents the development of mutated cells, prevents cancer or tumors. Oncogenes are cancer causing genes, and proto-oncogenes are normal genes that can become mutated. Then we come to meiosis. It's basically the basis of sexual reproduction. Only germ cells can undergo meiosis. So we have haploid cells, n equal to 23. Okay, the egg cell and the sperm cell are haploid cells, germ cells. Normal cells in the body are all 2n diploid, but the gametes are haploid, which get fertilized together to form a diploid zygote, 2n, which is 46, which can undergo mitosis to give rise to multicellular diploid adults. Okay, but these reproductive organs, here meiosis happens in order to get the haploid gametes out. Now, how meiosis halves the chromosome number? So, if you see at the beginning, there's a pair of homologous chromosomes in a diploid parent cell. Then the first is there are sister chromatids, a pair of duplicated homologous chromosomes after the S phase or the synthesis phase. Then meiosis 1 basically separates. There is no division of the centromere in case of meiosis 1. Okay, so if you see it is the same number basically. Meiosis 1 does not change the number, but then we have meiosis 2 where this happens. The number is reduced. So in the interphase, the chromosomes duplicate centromeres with centrioles, chromatin and nuclear envelope. But in case of the prophase one, these are the sites of crossing over, spindle. Sister chromatids are these are the fragments of the nuclear envelope. Okay, this is a tetrad formation, if you can see over here. The orange are the sister chromatids, the blue are sister chromatids, right? In metaphase one, the spindle microtubules get attached to the kinetochore. Okay, and then sister chromatids remain attached in the anaphase one and homologous chromosomes separate. In meiosis 2, the sister chromatids separate, right? So prophase 2, basically this. So in the first step, prophase 2, this happens. Then in metaphase 2, again a metaphasic plate or the eukaryotic plate is formed. In anaphase 2, the sister chromatids separate, and in telophase 2 cytokinesis, the haploid daughter cells are formed. Now there are three ways genetic diversity is increased by meiosis, right? So the two parents contribute one half of the genetic material to the offspring, right? Crossing over happens in prophase 1, chromosome alignment in metaphase 1. Meiosis produces sets that are not identical, okay, unique gametes. So then what is crossing over? What we talk about in the meiosis 1? So crossing over basically increases genetic diversity by producing new chromosomes. So if you see this is a tetrad, pair of homologous chromosomes in synapses. Oh, this is called a tetrad and this is called synapses regions, right? The breakage of the homologous chromatids will occur, then the joining of the homologous chromatids. If you see, there's a cross linkage happening. These points are known as chiasmatic points or chiasma. Then the separation of the homologous chromosomes will happen at anaphase 1. You see, there is a change, there's a exchange of genetic material, which is called crossing over. Then the separation of the chromatids at anaphase 2 and completion of meiosis will lead to gametes of 4 genetic types. This is a parental type of chromosome, all orange. This is a parental type of chromosome, all blue. These are the recombinant chromosomes. So independent orientation of chromosomes in meiosis and random fertilization lead to varied offspring. Okay. So independent orientation at metaphase 1, two equally probable arrangements of chromosomes in metaphase 1. Possibility A, possibility B, then metaphase 2, then gametes. This can be the combinations or this can be the combinations. Okay, so we have talked about cell cycle, mitosis and meiosis. So eukaryotic chromosomes contain DNA and protein and the chromosomes carry the genetic material. Talked about all of this. Okay, supercoil within the chromosomes, but basically these are DNA double helix, which are chromatins. Chromatin fiber is this. When a cell divides, chromatin fibers are very highly folded and become visible in the light microscope as chromosomes. During interphase, that is between divisions, chromatin is more extended, a form used for expressing a genetic information. The DNA is organized into informational units called genes, and chromosomes contain hundreds to thousands of genes. So the cell cycle, first the cell cycle is a sequence of cell growth and division, and the cell cycle is the period from the beginning of one division to the beginning of the next. So the time it takes to complete one cell is the cycle is generational time, doubling time or the generation time. So cells divide when they reach a certain stage. Okay. Cell division, like red blood cells and all of these cannot divide. Cell division involves mitosis and cytokinesis, which is basically karyokinesis and cytokinesis, together called mitosis. It involves the division of the chromosome. Cytokinesis involves division of the cytoplasm. So mitosis without cytokinesis can result in multinucleate cells. So if you imagine if the nucleus is divided but the cytokinesis is not happening, that will lead to one cell with multiple nuclei, right? Then the eukaryotic cell cycle is beginning of one division to the beginning of next. It is in eukaryotic cell cycle. Interphase is first gap phase, synthesis phase, second gap phase, M phase is mitosis, cytokinesis. This is the schematic of the cell cycle. You can see over here. Sorry for the animation. So this is the interphase. This entire region is interphase. And this we have the mitosis. So chromosomes become duplicated during interphase and cells are very active during interphase, synthesizing biological molecules and growing the G1 phase. Synthesis phase is marked by DNA replication and the G2 phase occurs between the S phase and mitosis. And despite differences between prokaryotes and eukaryotes, there are several common features in their cell division process. Like the replication of DNA is a must thing, right? They must occur. Segregation of the original and its replica follow, and cytokinesis ends the cell division process. So whether the cell was eukaryotic or prokaryotic, these basic events must occur. So hereditary material is passed on to the next generation by mitosis or meiosis. So chromosomal packaging of DNA allows efficient distribution of genetic materials during cell division. Life cycle requires two distinct types of cell division processes: mitosis and meiosis. One cell becomes two cells during an organism's life cycle. So mitosis is nuclear division plus cytokinesis and produces two identical daughter cells during the following steps: prophase, metaphase, anaphase, telophase. Interphase is often included in discussions of mitosis, but interphase is technically not part of mitosis, but rather encompasses stages G1, S, and G2 of the cell cycle. So in interphase, the cell is engaged in metabolic activity and performing it's prepared for mitosis. The next four phases that lead up to and include nuclear division. Chromosomes are not clearly discerned in the nucleus, although dark spot called the nucleolus may be visible, and the cell may contain a pair of centrioles or microtubule organizing centers in plants, both of which are organizational sites for microtubules. Now, prophase chromatin in the nucleus begins to condense and becomes visible in the light microscope as chromosomes, and the nucleolus disappears. So, centrioles begin moving to opposite ends of the cell, and fibers extend from the centromeres, and some fibers cross the cell to form mitotic spindle. And the nuclear membrane dissolves in prometaphase, marking the beginning of prometaphase, and proteins attach to the centromeres, creating the kinetochores basically, and microtubules are the kinetochores at the beginning of centromeres, and the microtubules attached at the kinetochores and the chromosomes begin moving. In metaphase, that is the most visible phase under the microscope, spindle fibers line the chromosomes along the middle of the cell nucleus. That line is referred to as the metaphase plate. Polar microtubules extend from the pole to the equator and typically overlap. Kinetochore microtubules extend from the pole to the kinetochores, and this organization helps to ensure that in the next phase, when the chromosomes are separated, each new nucleus will receive one copy of each chromosome. Now the anaphase, the paired chromosomes separate at the kinetochores and move to opposite sides of the cell, and the chromosomes are pulled by the kinetochore microtubules to the poles and form a V shape. And the motion results from a combination of kinetochore movement along the spindle microtubules and through the physical interaction of polar microtubules. Then near the telophase, the chromatids arrive at opposite poles of the cell and new membranes form around the outer nuclei, and the chromosomes disperse and are no longer visible under the light microscope. Spindle fibers disperse and cytokinesis will start. In animal cells, cytokinesis results when a fiber ring composed of a protein called actin around the center of the cell will contract, pinching the cell into two daughter cells, each with one nucleus. And in plant cells, this is a new cell wall between two daughter cells rather than the cleavage following cytoplasm. See images. Okay. So as you can see over here, in interphase, the cell is carrying out the normal life activities. Chromosomes become duplicated. Glias phase, early prophase nuclear envelope begins to disappear. Long fibers of chromatin become evident and begin to condense and visible chromosomes. In the late prophase, chromosomes may continue to shorten and thicken. Spindle forms between centrioles which have moved to the poles of the cell. Kinetochores begin attaching to microtubules. In metaphase, spindle fibers attach to the kinetochores of the chromosomes which line up along the cell's midplane. Chromatids separate at centromeres and one group of chromosomes move towards each pole. So chromosomes have arrived at the poles and the nuclear envelope begin to form. Cytokinesis produces two daughter cells, and interphase daughter cells formed are genetically identical to the parent cell. So there are images, if you can see. Very beautiful animation I have collected from somewhere. So reproduction essentially can be of two types, asexual and sexual. Asexual reproduction is a form of duplication using only mitosis. Like a new plant grows out of the root or a shoot from an existing plant, and it produces only genetically identical offspring since all divisions are by mitosis. But in sexual reproduction, it's formation of new individual by a combination of two haploid sex cells or gametes, and fertilization combination of the genetic information from two separate cells that have one half of the original genetic information. And gametes for fertilization usually come from the separate parents. So the female produces an egg, male produces sperm, and both gametes are haploid. With a single set of chromosomes, the new individual is called a zygote with two sets of chromosomes diploid, and meiosis is a process to convert a diploid cell to a haploid gamete, causing a change in the genetic information to increase diversity in the offspring. Now, chromosomes in a diploid set for humans 2 and 46. Autosomes will be homologous chromosomes, one from each parent. Sex chromosomes, humans have one set. Female sex chromosomes are homologous. Male sex chromosomes are non-homologous. Now, the number of sets of chromosomes in a cell, haploid is one set of chromosomes, diploid is two sets of chromosomes. Most plant and animal adults are diploid, and while as eggs and sperms are haploid. So, most cells in the human body are produced by mitosis. These are the somatic line cells. Cells that become gametes are referred to as germ-line cells. The vast majority of cell divisions in the human body are mitotic, with meiosis being restricted to the gonads. And in case of diploid cells, it's a characteristic number of chromosome pairs per cell. So it's homologous chromosomes are similar in length, shape, other features, and carry similar attributes. In haploid cells, they contain only one member of each homologous chromosome pair. Okay, you see over here. Then meiosis, diploid cells undergo meiosis to form haploid cells. Meiosis potentially produces four haploid cells. Meiosis involves two separate divisions. So two successive nuclear divisions occur. Meiosis 1 is reduction, meiosis 2 is division. So the number gets reduced, number of whether it will be n or 2n, that gets decided in meiosis 1 only. From 2n we get the n, and from meiosis 2 is basically similar, a replica of mitosis only. No change in number. So meiosis 1 reduces the ploidy level from 2n to n, as I said, reduction, while meiosis 2 divides the remaining set of chromosomes in a mitosis-like process, division. Exactly what I said. Then in meiosis, homologous chromosomes are separated into different daughter cells, whereas in meiosis 1 and meiosis 2, each included prophase, metaphase, anaphase, and telophase. In meiosis 1, prophase 1 is one of the most important stages of meiosis. During this stage, many crucial events occur. In prophase 1, spindle appears, nuclear envelopes disappear, DNA chromosomes begin to twist and condense, making the DNA visible to the microscope. Each chromosome actively seeks out its homologous pair, which also is sister chromatid. Then the two replicated homologous pairs will find each other and form a synapse, as I was saying, and the structure formed is referred to as the tetrad, which is the four chromatids. Now, the point at which the two non-sister chromatids intertwine is called a chiasma. Sometimes the process known as crossing over occurs at this point. Now, this is where two non-sister chromatids exchange genetic material. This exchange does not become evident, however, until the two homologous pairs separate. If you see the schematic over here, this exactly means that. Now, prophase 1 includes synapses and crossing over. Homologous chromosomes pair and undergo synapses. One member of the pair is the maternal homologue, the other is the paternal homologue, and synapses is the association of four chromatids, two from each homologue. And metaphase 1, the tetrads line up along the equator. Anaphase 1 results in the separation of homologous pairs, that is, cells are diploid at this point. And telophase 1 results in a brief reappearance of nuclear envelopes and the spindle disappears. Cell waits momentarily during interkinesis. It is basically a phase that separates meiosis 1 and 2, no DNA synthesis occurs. So in second division, that is meiosis 2, in prophase 2, the spindle reappears and the nuclear envelope fragments. In metaphase 2, the chromosomes align at the equator. In anaphase 2, sister chromatids separate. In telophase 2, the nuclear envelopes reappear and four haploid cells are the result. So these are the schematics. So germline cells undergo gametogenesis. Spermatogenesis produces sperm, and oogenesis typically produces eggs or a single ovum and two or more polar bodies. So there are three major stages to cell cycle: interphase, mitosis, and cytokinesis. Interphase encompasses the phases of G1, S, and G2. Mitosis encompasses the phases of prophase, metaphase, anaphase, and telophase. Cytokinesis is division of cytoplasm. So you see, this is the schematic. So why do we care about cell division? It's basically a process of reproducing cells, occurs during growth, repair, and development of tissues. The repeating sequence of cellular growth and division throughout the life of an organism is called cell cycle. So we have talked about all of these: growth phase. Cell grows rapidly and carries out routine functions. Phase is most of cell's life. Muscle and nerve cells never divide the remaining G1. Then S phase, cells DNA is copied. Synthesis phase, and then other stages. Chromosome consists of two chromatids attached to a centromere. Then we have the second growth phase of G2. Hollow microtubules are assembled. Microtubules are used to move chromosomes across during mitosis. In the second cycle phase of cycle, nucleus is divided into two nuclei. Each nuclei ends up with the same number of chromosomes as the original cell, includes prophase, metaphase, anaphase, and telophase. So in the final stage, cytoplasm divides. So there are three checkpoints. The G1 checkpoint basically decides when a cell can divide based on an environmental conditions, health, and cell size. Favorable conditions begin S phase. If not favorable, resting period begins. The G2 checkpoint is one DNA. Once DNA prepares enzymes and checks DNA replication. Once this checkpoint is passed, then mitosis can begin again. And another mitosis checkpoint signals end of mitosis and G1 begin again. So what happens when all checkpoints fail? So that is a good question. It can lead to cancer. Cancer is the uncontrolled growth of cells. Mutation missed by checkpoint can cause overproduction of growth hormone, and damage done to a cell by environmental factors can cause cells to constantly repair. So cell cycle is an ordered set of events that we have already discussed. The G1 phase stands for gap 1 and is required for cell growth and preparation of DNA synthesis. S phase stands for synthesis and replicates the DNA genome. G2 phase stands for gap 2 and is needed for cell growth and preparation for mitosis. Last phase is M, which stands for mitosis, where cells separate duplicated chromosomes. So in the G1 phase, the cell is doing its everyday job regardless of what type of cell it is. At this time, the chromosomes each have just one molecule of DNA. Okay, so chromosomes with one strand of DNA are called unduplicated or unreplicated chromosomes. So the gap 1 phase begins at the end of the mitosis and cytokinesis and lasts until the beginning of the S phase. So it is the longest 4 cell wall phase and varies in length. In the G1 phase, the cell grows and chooses to replicate its DNA or to exit the cell cycle and enter a quiescent state. During the G1 and G2 phases, cells grow and make sure that conditions are proper for DNA replication and their division. During this G1 phase, cells monitor the environment and determine if conditions, including the availability of nutrients, growth factors, and hormones, justify DNA replication. So this decision is to begin replication is made at a specific checkpoint in G1 called the restriction point. Now, if prior to the restriction point cells sense inadequate growth conditions or receive negative signals from other cells, they enter G0 phase, also called quiescence. So, in the G0 phase, they are maintained for prolonged periods in the non-dividing state. So, eukaryotic cell cannot divide unless it replaces DNA, that is genome, and then separates the duplicated DNA. Reduced cells must perform DNA synthesis and mitosis. So in the S phase, the DNA replicates or duplicates. The chromosomes that result have two molecules of DNA and are called duplicated or replicated chromosomes. And the S phase typically lasts about 6 hours. In mammalian cells, the start of S phase, when DNA synthesis begins, takes place several hours after the cell commitment to carry out the DNA synthesis. So the portion of interphase that follows S phase is called gap 2 phase. Some cells can exit the cell cycle from gap 2 phase just as they can from G1 phase. In G1 phase, the cell is carrying out processes necessary for mitosis to begin. As I was saying, in meiosis, it starts with 46 double-stranded chromosomes that is diploid or 2N. After first division, the reduction division, 23 double-stranded chromosomes are left in each cell. After second division, again 23 single-stranded chromosomes are left. So it occurs in our germ cells, cells that produce our gametes, egg and sperm. So meiosis 1 is the reduction phase, whereas meiosis 2 is the equational phase. Not to go into too much detail, here almost. Sorry. Back to the question that we started with, cell division occurs in the M phase of the cell cycle. Okay. So I'll wait for a few minutes here. If you have any doubts, you can tell me. Then I will go to the next one. 5 to 10 minutes. I'll wait.
Ma'am, ma'am,
Yeah, one second. Yeah.
actually, ma'am, I was having a doubt, well, not from this genetic engineering,
Yeah.
this
Yeah.
syllabus. Actually, I got the external doubt from my actual university.
Versatile.
We were, so, ma'am, we are working with the various stages of cooked potatoes like raw, half boiled, and full boiled. So when we are putting iodine on it, and after processing the image using the images software, the intensity by area, the raw potato is having the highest intensity by area, whereas in the case of boiled potato, it is very much lower than that. So why is it is happening means how?
I did not. Can you repeat what is higher in case of the intensity of what?
The stain, what the iodine I have put, I have taken an area, certain amount of area, same area for both that for raw potato, half boiled, and full boiled potato.
Okay.
So in that same amount of area, when I'm doing the intensity, in case of that raw one, it is very high intensity of that iodine,
Okay.
and when it
Yeah.
comes
Okay. So to the
to the
boiled
boiled one,
potato.
it's very,
So to the best of my
yes,
knowledge, a raw potato will have uh, a higher iodine stain. That's what you are saying, right?
yes,
Than a
ma'am.
boiled potato, because you know the iodine solution will turn blue black around when it reacts with starch, which is very much abundant in potatoes. So when a potato is cooked, the cell walls break down and the starch disperses. So the iodine stain will be much less intense. Okay, so raw potato will have a higher iodine stain than a boiled potato, because cooking a potato causes the starch to disperse, right? So the cell membrane, when you cook, basically when a potato is cooked, the cell membrane will probably get ruptured. The membrane around the vacuole will break, and the membrane around the starch will grain will also break. So the starch grain swells up, and the potato's texture becomes softer and swishier. So raw potato will have a higher iodine stain than a boiled one. Yeah.
Okay,
Okay.
ma'am.
Okay. Um, this, so this question, we can talk a little bit about this question. The difference between the TM of the forward and the reverse primer should not be more than A 10 degree, B 8 degree, C 2 degree, and D 12 degree Celsius. Which one do you think is right?
In option C.
Okay, so yeah, the answer will be 2 degrees Celsius. So if we talk a little bit about PCR, that's a polymerase chain reaction, which is an in vitro technique for the amplification of a region of DNA, which lies between the two regions of known sequence. PCR amplification is achieved by using oligonucleotide primers, and these are typically short, single-stranded oligonucleotides, which are complementary to the outer regions of known sequences. So the oligonucleotides serve as primers, basically, once again, as primers for DNA polymerase, and the denatured strands of the large DNA fragment serves as a template. So this results in the synthesis of new DNA strands which are complementary to the parent template strands, and these new strands are defined 5' ends. 5' ends are the oligonucleotide primers, whereas the 3' ends are potentially ambiguous in length. So this is how the PCR works. This is a template DNA. The first cycle will give rise to 4 copies, second cycle 8 copies, and so on and so forth. It's exponential amplification. Now, what are the steps of PCR? Step 1 will be denaturation. It is basically 94 degrees Celsius. Temperature will be 94 degrees Celsius. Then we have annealing. It will be 54 degrees Celsius for forward and reverse primers. And then we have extension. We have 72 degrees Celsius only DNTPs. Now, PCR contains target DNA, for example, the environmental DNA, 2 primers which are 20 to 30 nucleotides long, thermostable DNA polymerases, then nucleotides and the magnesium ion, which is a cofactor for the DNA polymerase. Okay, so each cycle will go through the process of denaturation, annealing, and extension. So the temperature should be adjusted to balance between specificity and amplification, right? And this is the schematic of each cycle. Now, if we talk about primers, how do we basically design or select primers? So primer selection, primer is an oligonucleotide sequence which will target a specific sequence of opposite base pairing like ATGC only of single-stranded nucleic acids. For example, there is a 1/4 chance of finding an AGC or T in a given DNA sequence. There is a 1/6 chance of finding an indinucleotide sequence and a 1/56 chance of finding a given 4 base sequence. Thus, a 16 base sequence will statistically be present only once in every 416 bases. So this is about the size of the human or maize genome and 1000x greater than the genome size of E. coli. So primer specificity, universal means amplifies all bacterial DNA, for instance. Group specific means amplify all denitrifiers, for instance. Specific means amplifies just a given sequence. Now for forward and reverse primers, if you know the sequence targeted for amplification, you know the size with the primer should be annealing across. And if you don't know the sequence, what do you get basically? What is DNA polymerase? It's an enzyme responsible for copying the sequence starting at the primer from the single strand. Now commonly used is stacked DNA polymerase, an enzyme from the hyperthermophilic organisms Thermus aquaticus, isolated first at a thermal spring in Yellowstone National Park. So this enzyme is a heat tolerant, useful both because it is thermally tolerant. It survives the melting temperature of DNA denaturation, which also means the process is more specific. Higher temperatures result in less mismatch, more specific replication. Okay. You need to see all this. So it was invented in 1983 by Gary Mullis, who won a Nobel Prize, and this technique basically made possible the discovery of Taq polymerase, the DNA polymerase that is used in the bacteria Thermus aquaticus, discovered in hot springs. The DNA nucleotides are the building blocks of the new DNA. Template DNA is the DNA sequence that you want to amplify. Primers, single-stranded short DNA that are complementary to a short region on either end of the template DNA. And DNA polymerase is a heat-stable enzyme that catalyzes the synthesis of the new DNA. Okay, so how do you start designing the primers? So this primers can be designed by several different softwares, and different softwares may vary significantly in different things, right? So what are the general rules that should be followed for primer design? Primer length determines the specificity and significantly affect its annealing to the template. So too short will be low specificity, resulting in non-specific amplification. Too long will decrease the template binding efficiency at normal annealing temperature due to the higher probability of forming secondary structures such as hairpins. Then we have the optimal primer length. It is 18 to 24 base pairs for general applications, 30 to 35 base pairs for multiplex PCR, Then you have the optimal amplicon size, which is 300 to 1000 BP for base pair for general applications. You should avoid greater than 3 kilobases, and 50 to 150 BP for base pairs for real-time PCR. You should avoid greater than 400 base pairs. Now, what are the general rules for melting temperature? It's the temperature at which 50% of the DNA duplex dissociates to become single-stranded. Is determined by primer length, base composition, and concentration. It's also affected by the salt concentration of the PCR reaction mix. So, working approximation will be Tm is 2A plus T plus 4G plus C, suitable only for 18 bar or shorter. Optical melting temperature 52 degrees Celsius to 60 degrees Celsius. Melting temperature above 65 degrees Celsius could be generally avoided because of the potential for secondary annealing. Higher melting temperature is recommended for amplifying high GC content targets. Primer pair TM mismatch, significant primer pair melting temperature dispatch can lead to poor amplification. So the desirable TM difference should be lesser than 55 degree between the primer pair. Should not be more than 5 degree between the primer pair, right? Then obviously specificity, so determined primarily by primer length as well as sequence. Adequacy of primer specificity is dependent on the nature of the template used in PCR reaction. Cross homology may become a problem when PCR template is genomic DNA or consists of mixed gene fragments. Primers containing highly repetitive sequences are prone to generate new non-specific amplicons when amplifying genomic DNA. Avoid non-specific amplification, like you can blast as a software basically to see the similarity, the name same nucleotides. So blasting PCR primers against NCBI non-redundant sequence database is a common way to avoid designing primers that may amplify non-targeted homologous regions, and primers spanning intron exon boundaries to avoid non-specific amplification of gDNA due to cDNA contamination. So primers spanning exon boundaries to avoid non-specific amplification cDNA due to gDNA contamination. Okay, so GC content of primer should be around 45 to 55 degrees Celsius, 55%. Common GC content is 40 to 60%. Long runs increases mispriming, non-specific annealing potential. Maximum acceptable number of runs is 4 base pairs. Repeats increases mispriming potential. The maximum acceptable number of repeats is 4 dinucleotide. Okay. GC clamp, so it refers to the presence of G or C within the last 4 bases from the 3' end of primers. Is essential for preventing mispriming and enhancing specific primer temperature binding. So we should avoid greater than 3 guanines or cytosines near the 3' end. Then you have the max 3' end stability. It refers to the maximum delta G of 5 bases from the 3' end of the primers. So while higher 3' end stability improves priming efficiency, too high stability could negatively affect specificity because of the 3' terminal partial hybridization induced non-specific extension. Some of the softwares that people use for designing primers. Okay, so as we know, in summary, denaturation scene of PCR will be 95 degrees Celsius, which basically means the double-stranded template DNA will be denatured by heating, typically to 25 degrees Celsius, to separate the double-stranded DNA. Next is annealing, could be one of the temperature 50 to 65 degrees Celsius. Reaction is rapidly cooled to an annealing temperature to allow the oligonucleotide primers to hybridize to the template. It anneals only to sequences that are complementary to them. Then extension is 70 degrees Celsius. The reaction is heated to a temperature typically 70 degrees Celsius for efficient DNA synthesis by the thermostable DNA polymerase. Basically, an artificial way of doing DNA replication. We know it involves melting DNA, priming, polymerization. This is schematic. So what are the components of PCR reaction? The buffer, which contains magnesium, template DNA, two primers that flank the fragment of DNA to be amplified, DNTPs, tag DNA polymerase. You see, this is the melting, which, which happens at a higher temperature. DNA strands are denatured and separate from each other. Then annealing, where the primers anneal. Then extension, where the primers extend. Sorry. So theoretical yield of PCR, if you start with 100 copies, how many copies are made in 30 cycles? It's this value. So you can see how many copies of DNA can get made in 30 cycles. That was coming back to the so we're coming back to the question that we started with. The difference between the TMF forward and the reverse primer should not be more than 2 degrees Celsius, right? So if you have a doubt regarding this question, you can ask, or we can move on to the next question. Okay, so the next question is the correct sequence of steps during plasmid isolation by alkaline lysis is. So there are 4 options. You can try answering this question. I'll wait for a few minutes, then we'll discuss. Tell me which one is the correct option.
Option B.
Option B or D?
B.
Which one? Can you read out the
Alkaline lysis, renaturation, deproteination, and alcohol precipitation.
So yes, the correct answer is option B. That is the correct sequence of steps during plasmid isolation by alkaline lysis. So when we are doing plasmid isolation in labs, basically alkaline lysis is the easiest technique because there are a bunch of things that you can make in lab. The chemicals, the reagents you can make, add, mix a number of things with the proper ratio and can make in lab, and you can use this process. So what is the concept of alkaline lysis and what are the steps involved? So isolation plasmids are, as we know, are extra chromosomal DNA structures found in bacteria and some yeast, which do not encode vital genes, but which can help the survival of microorganisms in various antibiotics, in the presence of various antibiotics and all of that, right? So plasmids are very important in molecular biology because they are frequently used in cloning studies, right? So the basis of alkaline lysis method is that pH changes in the environment have a different effect on chromosomal and plasmid origin DNA fragments. So basically, when the medium becomes alkaline, both chromosomal and plasmid DNA become a single strand. However, when the pH of the medium is lowered and acidified again, the strands of the small plasmid DNA can merge rapidly, but since the chromosomal DNA is larger, the double chain formation does not occur easily and the chromosomal DNA is denatured. So in this way, chromosomal DNA and plasmid DNA can be separated. So what happens basically, it's a schematic. We take a single bacterial colony grown in Luria botany LB broth in a medium of antibiotics shaking oven at 60 degrees overnight, and we take that. We basically separate the media from the pelleted cells, and then we do the process of alkaline lysis. So first we add solution 1, which basically contains NaOH. Solution 1, which basically tell you what it contains over here. Solution 1 contains glucose tris EDTA. Then we add the another solution which is NaOH and STS, and at the end we add potassium acetate glacial acetic acid. So what happens? The EDTA disrupts the cell structure by binding Mg2 plus ions, which are necessary for the protection of the cell structure, and inhibits the cell enzymes that enable the degradation of DNA. Then STS removes lipid molecules by lysis and disrupts the cell membrane structure. Phenylchloroform precipitates the proteins and removes them from the DNA, whereas pure ethanol competes with the water molecules in the medium to allow the DNA to collapse. 70% ethanol, sodium such as monovalent cations and salts to dissolve away from the environment. Okay. So basically we need to eliminate membranes, proteins, chromosomal DNA and like genome and RNA. So basically we lyse cells, it rupture the membranes typically with alkaline lysis. High pH and detergents will lyse the cells. Then neutralized pH will cause the membranes to clump. Then chromosomal DNA stays attached to the membranes unless it is sheared by rough pipetting or vortexing, and we can centrifuge to pellet the membrane chromosome complex. Now, alkaline lysis solutions also contain RNAs which degrade the RNA rapidly. Okay. Now we are left with the proteins and plasmid DNA in solution. So you can load on a spin column with nylon membrane. So DNA will bind to nylon. Presence of alcohol will strengthen this. Now proteins will spin through membrane in the presence of alcohol. After this step, only plasmid DNA should be left on membrane. So you can add water or buffer to let the plasmid DNA leave membrane and spin into a fresh tube. So this is the basic concept. So this can play out in various ways with different steps. Okay, cells can be lysed by putting cells with lysozyme, also then putting in boiling water, then removing membranes and chromosomes by centrifugation. RNA is degraded by RNAs. Proteins are removed by phenol extraction. Phenol is nonpolar, so proteins will tend to collect at polar-nonpolar interface of extraction. We can collect the aqueous phase, which should have plasmid DNA. Precipitate will get the precipitated plasmid DNA. That DNA will precipitate in cold ethanol with sodium chloride present, and the pelleted precipitated plasmid DNA by centrifugation, and the dry and resuspend. The pellet can be dried and resuspended in water or buffer. So in order to isolate the chromosomal DNA, we obviously will lyse the cells with detergent at neutral pH, and the chromosome will not adhere to the membranes. We have to degrade the RNA with RNase, remove proteins with proteinase or by phenol extraction, and chromosomal DNA will make long strands and precipitate in isopropanol. Plasmids will not make strands, so hook-stranded DNA form precipitation and resuspended. If you want to isolate the RNA, we have to lyse the cells, remove membranes by centrifugation, degrade DNA with DNase enzyme, remove proteins by protease or phenol extraction, and precipitate RNA in a very cold ethanol precipitation or on column. If you want to isolate the proteins, we have to lyse the cells and remove membranes by centrifugation, degrade RNA with RNase, degrade DNA with DNase, precipitate protein with ammonium sulfate, and suspend and dialyze to remove excess salt. Okay. You know what plasmid is? So what basically plasmids are small double-stranded extrachromosomal DNA molecules, right? They can be isolated from bacterial cells. They replicate independently of the bacterial chromosome. They can be found in a wide variety of bacterial extrachromosome bacteria. They are extrachromosomal elements which replicate independently of the bacterial chromosome, and they are not essential for the bacteria but may confer a selective advantage. So using the enzymes and proteins encoded by their host for their interplication and transcription, transcription. They can be inherited and they can be used in many applications. Now, there are three general classes of plasmids which can be advantageous for a host cell. Virulence plasmids encoding toxic genes that turn the bacteria into a pathogen. Drug resistant plasmids that confer resistance to antibiotics containing antibiotic or drug resistant gene. And plasmids encode gene required for bacterial conjugation. So in case of the plasmid copy number, it is one of the most critical factors affecting the yield of plasmid from a given system. It is determined primarily by the replicon. Replicon is the region of DNA surrounding and including the region of replication in the plasmid. So usually low copy number yield is 25 copies of the plasmid per bacterial cell. High copy number plasmid yield is approximately 200 to 700 plasmid copies per cell. So plasmid applications are important tools in genetics and biochemistry labs. They are commonly used to multiply or express particular genes, gene therapy, molecular cloning, and plasmids which are used in genetic engineering are called vectors. This is how a plasmid map looks like. So they'll have not a very huge role to play, but they might have some antibiotic resistant genes that will give them a selective advantage, right? So what are the different components in this plasmid map? The ORI, or the origin of replication, which basically is a DNA sequence that allows the initiation of replication within a plasmid by recruiting transcriptional machinery proteins. Then we have antibiotic resistant gene. It allows for selection of plasmid containing bacteria. Then we have MCS, or multiple cloning site. So it's a short segment of DNA which contains several restriction sites, allowing for the easy insertion of DNA. In expression plasmids, the MCS is often downstream from the promoter. Then we have the insert, is the gene insert, gene promoter or other DNA fragment cloned into the MCS for further study. Then we have the promoter region, drives transcription of the target gene. Vital component for the expression vectors determines which cell types the gene is expressed in and the amount of recombinant protein obtained. We have the selectable marker. There is the antibiotic resistance gene that allows for selection in bacteria. However, many plasmids also have selectable markers for use in other cell types. Then we have the primer binding site, a short single-stranded DNA sequence used as an initiation point for PCR amplification or sequencing. So primers can be exploited for sequence verification of plasmids. This is a schematic outline. You need to harvest cells by centrifugation, basically pellet down the cells and remove the supernatant, which is the media. We suspend the cell pellet in detergent and neutralize with QAC, and then plasmid and protein and RNA, everything will be here. Take aqueous ethanol precipitate and we'll get the pure plasmid. So depending upon nutritional status, bacteria exhibit different growth patterns which include, as I discussed in one of the previous classes, lag phase, which is in this phase the bacteria is still getting accustomed to the new environment. They adapt themselves into growth conditions and synthesize its own DNA, RNA proteins. Then we have the log or the exponential, the most healthy phase, where the bacterial cells are healthy, happily dividing, and the production of new cells is proportional to increased time. Then we have the stationary phase, the growth rate slows as nutrients become limited, waste products accumulate, and the rate of cell division equals the rate of death. Then we have the death phase, due to continuous accumulation of toxic metabolites and the lack of nutrients, death occurs of the bacteria. So we use principle of the experiment, basically using SDS is an alkaline solution. SDS will lyse the bacterial cell membrane and denature the proteins too, and the alkaline pH will denature the genomic DNA and denature the proteins too. The degraded cell wall, denatured chromosomal DNA and bacterial proteins will form large aggregated complex which will precipitate and be removed by centrifugation, whereas the native plasmid DNA can be collected from the supernatant. So coming back to the question that we started with, the correct sequence of steps during plasmid isolation by alkaline lysis is alkaline lysis, renaturation, deproteination, and alcohol precipitation. So you're right. Okay. If you have any questions regarding this, queries regarding this question, we can discuss, or we can conclude the session for today. Anybody? Okay, so if you have not have any questions regarding this, we can maybe stop the session today, and we can meet again in the next session, that is next Tuesday, and we can conclude the session for today. Thank you.
Thank you. Thank you, ma'am.
And a decent pair of shoes, maybe a shirt.